We can see it, Sharnom.
Okay, got it. All right. I'll start sharing right now. All right, so first we'll be going over chapter 38, which goes over homeostasis, temperature regulation, etc. So why do humans and other animals exhibit homeostasis? So first, it's important to know about the structure of our overall human body. Um, well, obviously we have atom, molecules, cells, and then this goes into tissues. These tissues form organs, organs form the organ system, and then eventually this forms organisms, which is us. So there are multiple tissue types, and there are four main tissue types that we're going to talk about. So the four main tissue types are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So there's three types of epithelial tissue. There's simple, stratified, and pseudostratified. There's also connective tissue. Connective tissue is primarily found in our bones, bloods. There's also in our areolas. There's also two other types of connective tissues, which are dense regular and dense irregular, and then more. Our muscle tissues, which are skeletal muscle, smooth muscle, as well as our cardiac muscle. And then lastly, we have our nervous tissue, which makes up our neurons, glia, etc. So for epithelial tissue, like I mentioned, there's a lot of different types of epithelial tissue, which each have their own usages. So simple squamous, which is found in our lungs and also air sacs, blood vessels, etc., they help with the exchange of any nutrients, wastes, or gases. Stratified squamous, which is the outer layer of the skin, mouth, and vagina, they help protect against abrasion or infection. There's simple cuboidal and stratified cuboidal. So simple cuboidal lines the kidney, tubules, and glands, and they help with reabsorption and secretion of water. And then stratified cuboidal, which lines the ducts of sweat glands, and they also help with secreting water and other ions. Lastly, there's simple columnar and stratified columnar. So simple columnar is in most digestive organs. They help produce mucus. So mucus is important because it helps prevent any extra acid or anything negative that could get absorbed in our body. It also helps with absorbing nutrients. And then stratified columnar helps line the epididymis, mammary glands, and the larynx, and it also helps secrete mucus. And also, if anyone has any questions, just like raise your hand and you could ask in the middle if you want me to go over something anymore. So epithelial tissues, I'm sure maybe you've seen them during your lab, but they create compartments in and of themselves. So for example, for stratified squamous, they help create our skin. So they help protect the body. And as you can see, there's lots of layers within them. Cuboidal epithelium make up ducts, and they also have absorption functions. Um, the stomach lining also includes epithelial cells that help secrete acid or digestive juices in order to help with absorption later on. Um, and then we also have some epithelium cells that line the internal organs, such as the airways of the lungs and the small intestines, and they also have secretory and absorption functions. Uh-huh. So, muscle tissue, we have three types of main muscle tissue: cardiac muscle, skeletal muscle, and smooth muscle. So, cardiac muscle helps with the contractions of the heart. Skeletal muscle helps with movements of your actual body itself. And then smooth muscle provides motility to internal organs such as the digestive tract. So, it helps with transferring food or other molecules, etc., that's in your digestive tract to go onwards. And it also controls the diameter of blood vessels to help blood or other molecules go through faster, quicker, or slower even. Connective tissue, we have lots of different types of connective tissue. So example, connective tissue is your bones. So your bone helps provide support for your body. That obviously makes it possible for your muscles to move. You have blood cells. Red blood cells carry oxygen that'll help obviously oxygen is imported throughout your body. Red blood cells help transport it throughout your body. And then white blood cells are the protectors of the cell, protect against any foreign bodies or microorganisms that are within your body. Adipose tissue is like fat, so they help support the organs, they help provide any thermal protection, and they help store energy as well. And then lastly, you have your ligaments and tendons, which connect your bones to other bones and then connect your muscle to the bones as well. Furthermore, there's nervous tissue. So nervous tissue is part of your brain, sensory system, etc. So there are sensors such as how the cells of the retina encode information about the sensory environment. So obviously you have your brain and the spinal cord, but then you also have your peripheral nervous system, which is everywhere throughout your body. So obviously you need sensors to help detect any stimuli from the environment. You also have neurons, which communicate information from these sensors to the central nervous system, and then they help store and integrate this information. And then you also have glia, which support your neurons, such as insulation and providing immune function for the central nervous system as well. So this is the structure of your nervous tissue. So we'll go over this later on for the nervous system chapter as well. So this is just going to be like a brief overview. You have your neuron here, you have axons here. Your axon is where action potentials happen, which we'll also discuss later. And then these are other neurons, etc., that help. All the neurons are connected to one another. So these neurons connect to other neurons and they help send information throughout your body. So obviously, in order to have your organ system, as we described, or your organ system starts from an atom and then obviously works its way up to an organ system and currently what humans are as well. So different types of tissues have to interact with one another in order to make organs, and they usually consist of all four tissue types. So for example, in the stomach, you have muscle to help move food through, you have muscle tissue to move food through the gut. You have epithelial tissue, which help cover the organs and line the cavity, and they also produce mucus as well. Then they also secrete juices, secrete hormones, some absorb nutrients as well. There's also nervous tissue to help coordinate any contractions of the smooth muscle and provide sensory function for the gut. So either way, all your organs are a combination of several different types of tissues. They all need the help of these tissues work its way up, in order to work its way up towards what humans are today. So your body also has an internal environment that it needs to keep. So homeostasis. So your body obviously needs stability. In terms of stability, your body wants to remain at a regular temperature, regular everything. Everything wants to be as stable as possible. Our body doesn't necessarily like any change. There's also a set point. So the set point is the point where your body is essentially at its most stable. You have sensors throughout your body in order to detect any stimuli from the environment in order to adapt to any change within the environment. You have regulators. Regulators obviously regulate what's going on in your body, also to help adapt and make sure that nothing happens immensely in your body. Because once again, the main thing about our body or humans in general is stability. And then you have feedback loops. So feedback loops are basically in your body. Once a change happens, feedback loops help determine how much of that change should happen. So there's positive feedback loops, negative feedback loops. They help either strengthen the effect of the change or they negate the effect of the change. They go back into the system and then tell the body to stop causing the change. So feedback loops are very important parts of human bodies in order to help determine what should happen. So negative feedback loops are stabilizing, positive feedback loops are destabilizing because once again, negative feedback loops help prevent any extra change from occurring, while positive feedback loops cause extra stimuli and cause the change to multiply essentially. So you have an external environment and an internal environment. So your external environment changes obviously depending on where you are, location, etc. And it's also a source of energy and nutrients. And it's also a repository of waste. While your internal environment is, oh, sorry. While your internal environment, like I mentioned, likes to remain as stable as possible. Um, likes to remain as stable as possible. It responds to changes in the external. Oh, sorry. It responds to changes in the external and both the internal environment. And I received a chat. Someone asked if they could help explain feed forward information. So let me just go back. So once again, when it comes to feedback loops, we'll talk about feedback loops in other chapters as well. Feedback loops are a critical part of the human body system. So once again, there are negative feedback loops, which is essentially when your body notices a change in the environment or within the environment, it could create a feedback loop. So it essentially circles around. So it could tell an organ, for example, to stop releasing any hormones in order to amplify that change because obviously your body wants to remain stable. So it's a all everything within our body is a loop. So our body notices a change and then our body tries to adapt to that change. So there's a stimuli present and our body starts to adapt. But obviously we don't want too much change to occur. So there could be a feedback loop where it goes back around and it tells our body to stop producing it tells an organ of the body to stop producing hormones or stop trying to amplify that change. While positive feedback loops, essentially, it just keeps on stimulating that organ or stimulating the body to keep producing hormones and keep producing changes within that body. And we'll talk about this more because it's feedback loops are an important part of pretty much all your systems within your body. Set point. So set point is the point where your body, sorry, so, people are sending private chat, so I just wanted to go over that. So set point is basically the point in your body where your body wants to remain the most stable and return to at the end. It's like the it's like that one point where your body is at its most stable, and that's what it likes to remain at. So thermal regulation. Uh, in within your body likes to remain at a certain temperature. So 37 degrees here is the normal zone. That could be considered like your set point for your human temperature. And as you go in a hotter temperature within, that's why they say if you have an extremely high fever, like if you're over 105 degrees Fahrenheit, make sure you definitely visit a doctor, visit someone because prolonged time at such a high uh at a high temperature can cause tissue damage, seizures, or even brain cell death. And then even lower temperatures as well, confusion, slow reaction, and if you go extremely low, you could also have brain damage. So you want your body to remain as stable as possible, which in this case is around 37 degrees Celsius. So there's lots of strategies for how our body regulates our temperature throughout the body. Um, so there are ectotherms and endotherms and heterotherms. So ectotherms, body temperature depends on an external source of heat. So your body temperature follows the temperature that's outside the environment. There are endotherms, which are temperature regulators. So your body temperature is maintained at a preset temperature. So that's humans. Our body temperature likes to remain at that 37 degrees Celsius limit. Um, and then there's heterotherms. Now, heterotherms aren't as common. However, their body temperatures are maintained at preset temperatures under some conditions, and depending on other conditions, they follow temperatures from the external environment. So heterotherms it switches depending on the condition they're under. So metabolic rate is inversely proportional to body mass. So what this means is, um, so you have a shrew. Shrew obviously has a small body mass. So animals with a small body mass have a small surface area. As a result, they extend a lot of heat throughout their body. They release a lot of heat throughout their body. Now, your body does now, even for shrews or even for other animals, you don't want them to release heat as much throughout their body. You want them to maintain heat as much as possible. So as a result, the metabolic rate increases in order to help cover up for that heat loss. So once again, for shrews, mouses, etc., because they're so small, they release so much heat. Since they release so much heat, the metabolic rate has to increase as well in order to help stabilize it so that the animal could maintain its body temperature. So thermoregulation and ectotherms. So once again, ectotherms are animals that depend on the outside environment. So there's behavioral adjustments. Some animals could shelter or bask in the sunlight to maintain body temperature. There are also biochemical or metabolic adjustments they could do within their body. Um, there are isozymes to help keep their metabolic rate constant. There are also physiological adjustments as well. So they could redirect the blood flow within their body to conserve heat and change heart rate. And there's also anatomical adaptations as well. So, for example, there are types of fish, depending on the environment they're in, they could modify the vasculature within their own body in order to help conserve heat. So as you can see here, the cold fish and the hot fish, they have completely different vasculature, but they both help maintain conserve heat depending on obviously the environment they're in. So there's lots of ways that ectotherms do adapt to their environment in order to maintain heat. It's not as if they die really quickly or depending on their environment or they always live in like one certain temperature. They manage to adapt to it. So the mammalian thermoregulatory systems. So for humans, we shiver, we sweat, we salivate, we pant. That all either produces heat or releases heat. Then we also have heat conservations within us, behavioral, postural adjustments, whether we like hug ourselves, etc. Fat deposits, fur in other animals, wool, feathers, etc., which help all increase insulation. And then also our vasculature as well. So even mammals have different ways to maintain temperatures. So metabolic rate is ambient temperature dependent. And what I mean by that is, so the basal metabolic rate is the rate required to maintain minimal body function. And then there's a thermal neutral zone. So the thermal neutral zone is the ambient temperature or the external temperature at which the basal metabolic rate generates enough heat in order to maintain your actual body temperature. And then you have a lower critical temperature and an upper critical temperature. So a lower critical temperature is when or upper critical temperature increases in metabolic rates is necessary in order to either produce heat or lower heat. So obviously, the lower the temperature, the higher the metabolic rate is necessary in order to produce heat, because you obviously want your internal body to get hotter. And then the opposite is also true. If you go above the upper critical temperature, then you want your body to increase the metabolic rate, but in the opposite direction in order to increase heat loss. So you'd start sweating, etc. Start trying to release heat as much as possible. So that was chapter 38. I also wanted to give like a little I just wanted to say that there are a lot of material that I'm not going over. I'm trying to go over the major concepts of each chapter. Because obviously this is only 2 hours and we're trying to go over every chapter. So if I don't go over something, that doesn't necessarily mean that Dr. Garofalo isn't going to go over it. So after I'm done with the chapters, I'm perfectly happy to answer any general questions you have either about the chapters, study tips, etc. as well. So just wanted to tell you guys that if I'm not just because I'm not going over something doesn't mean that it's not going to be on the exam. So these are just like the main ideas or main concepts from each chapter. So we're moving on to chapter 39 now, which is animal hormones. So the endocrine system, which is responsible for pretty much the majority of hormones throughout our body. So it's a complex network of glands and organs, and it helps control several aspects of our body from metabolism, reproduction, response to injuries, our mood, growth, etc. So what is a hormone? So hormones are regulatory substances produced within organisms, and they're the chemical messengers that help tell other organs what to do. So they have specialized receptors in order to communicate their message to the body. So one thing that's important is that each hormone essentially has a specialized receptor that only that hormone could bind to. So that's how our body makes sure that other hormones can't affect another body system. Each hormone has their own specialized receptors in order to help communicate their message to the body. So hormones are transported in various ways. They're transported in fluid, such as blood or sap, to stimulate specific tissue into action. There's also distant and local transport. And then hormones also influence several things, and then they work slowly over time and they affect several functions, like I mentioned, throughout our body, from energy, reproduction, mood. You know? And they also are important for growth, development, metabolism too. So autocrine versus paracrine signaling. So signals that act at a distance. So this is distant transport. So you have cells that secrete hormones, and these hormones travel through the blood and then manage to bind to receptors on target cells in the distance. Um, and then autocrine signaling on the other hand. So autocrine and paracrine signals are local signals. So autocrine signals bind to receptors on the same cell that secrete them. So for example, here you have a signaling cell. These blue circles are hormones that are released. So they release these hormones, but they bind to a receptor within that same cell. So once again, autocrine signals bind to the same cell that secreted the receptors, while paracrine signals bind to receptors on nearby cells. So this target cell has no receptors, so these hormones can't bind to the cell. However, this target cell does have a receptor, so the hormone can bind to the cell, causing a signal to occur. So once again, signals act at a distance, and for locally, there's two types, autocrine and paracrine. Autocrine is within the same cell, paracrine is nearby cells. So there are three classes of hormones. There's peptide and protein hormones. For example, insulin. They're water soluble, so they could be easily transported throughout the blood. There's steroid hormones, such as testosterone and estrogen. They're lipid soluble, and they're also able to pass through the cell membrane thanks to the phospholipid bilayer. And there's amine hormones. So amine hormones such as epinephrine, thyroxine, and they could be both water and lipid soluble. It depends on the type of hormone, how large it is, etc. And so you don't necessarily need to memorize obviously all of this, but this is just the different types of hormones. Obviously, protein hormones are made up of amino acids, steroid hormones are lipid soluble, and then you have amine hormones at the bottom. Fight or flight response. So the fight or flight response is really important. So the brain detects any danger from the outside environment, and it signals your adrenal glands to help release epinephrine and norepinephrine to trigger a number of effects. So in this case, the heart starts beating faster, obviously, so your blood pressure rises, blood is transported throughout the rest of your body. The liver breaks down any glycogen to supply glucose to the blood, obviously, to help provide more energy. You need more ATP in your body at this current point. Fat cells also help release any fatty acids and fuel to the blood. And then blood vessels in your skin contract to help shunt more blood to the muscles. So all of this is responsible for your fight response or your flight response. Whichever one you do, they both are caused by the same processes. So prolactin is a conserved structure, but it's an evolved function. So prolactin is interesting because as organisms diversified, the same hormone and the same receptor type of systems diversified itself depending on the type of organism to serve different functions, even though it's all the same hormone at the end of the day. Oh, sorry, someone put a question. If they are water soluble, doesn't that automatically mean it's lipid soluble? Not necessarily, because if they are water soluble, um, So lipids do contain water, but obviously if something you can't, if you can't do it vice versa, for example. If something is water soluble, that doesn't necessarily mean it's lipid soluble because it has to be able to transport throughout the body. So if something is water soluble, it has to be transport throughout the liquid, such as, for example, your blood throughout the body. That doesn't necessarily mean it could go through the phospholipid bilayer. So you can't have it like vice versa, for example. And also feel free to raise your hand if you guys have questions. So when it comes to prolactin, so prolactin is different in all types of animals. Um, so in fish, prolactin is required for osmoregulation. In freshwater species, and in saltwater species, it helps return of animals such as salmon in or to their spawn. Because as you know, salmon, they release their eggs, then they swim away, and then eventually, when it comes time for them to return back to their habitats in order to release their eggs again, prolactin helps cause that. The same thing happens in amphibians, where prolactin creates a water drive to help return adults to their breeding locations. And also, it stimulates lots of development of the oviducts, egg jellies, etc. While in mammals, prolactin helps stimulate growth of the mammary glands and also milk productions. So, the same hormone and the same sensory systems in three different organisms um causes three completely different functions. So, I mean, that's one of the major things about prolactin. It's conserved, but it evolved its functions depending on the type of organism it remained within. So now we're going to talk about the endocrine system of humans. So this is obviously a lot of what is going on within your body. So your hypothalamus is where hormones released from the posterior pituitary gland and hormones regulate the anterior pituitary gland. So when it comes to memorization, obviously these are a lot of hormones. However, try your best to memorize as many hormones as possible, or at least the major hormones, because a lot of these hormones are responsible in a lot of your other systems as well. For example, later on when we talk about animal reproduction, um, you're going to learn about oxytocin, follicle stimulating hormone, and luteinizing hormone. They're all extremely important, and they're all from the pituitary gland. And then also you have growth hormone, stimulates growth, metabolic functions within your body. You also have um prolactin, stimulates milk production and secretion, which we just talked about. ADH, thyroid gland also has lots of hormones. So there are a lot of hormones that you need to memorize. Like as you can see here, it seems like a lot. However, a lot of these hormones are responsible for other systems in your body. So as long as you memorize them either in this chapter or other chapters, you'll still be good overall. And then obviously this is even more. Now we're talking about insulin, glucagon, epinephrine, etc. So once again, these there are a lot of hormones. However, you'll go over the majority of them as you go through each individual system. So just as long as you know about it beforehand, it'll help out. Someone put a question. How can we distinct between the posterior and anterior gland? So, anterior. So as you can see here, the pituitary gland and anterior pituitary gland. Just think of it as the pituitary gland as a whole. The actual difference between the anterior pituitary and the posterior pituitary, um, as long as you know for the most part that it's produced within the pituitary gland, you should be fine. So now we're going to talk about the brain pituitary gland axis. So the human pituitary gland secretes many hormones, but it's extremely small. However, it does secrete many hormones. So the hypothalamus, which is also in your brain, it produces ADH and oxytocin, and then transports them to the posterior pituitary. And then neurohormones are then released by nerve terminals in the posterior pituitary and then diffuse into capillaries. And then capillaries are part of your blood, and then they leave the posterior pituitary via the blood to go to whichever organ it needs to go to. So now we're going to talk about positive and negative feedback loops, which I discussed before, but now we're going to talk about it in a bit more detail. So there are two hierarchical superimposing pathways. You have a stimulatory pathway, which is your positive feedback loop, and they also have a feedback inhibitory pathway. So for example, in this example here, the green arrows are stimulation. Hypothalamus releases hormones, which stimulates the anterior pituitary. The anterior pituitary produces tropic hormones, which then stimulates the endocrine gland, which then stimulates the endocrine gland to produce more hormones. So it's a straight loop, it's a straight step of stimulation. However, there's also inhibition in each step. Some of the hormones released by the endocrine gland can go back to inhibit the anterior pituitary to help prevent it from being stimulated, or it could even go back all the way to the hypothalamus to prevent it from being stimulated. And that's the long loop because after several steps, that hormone can go all the way back to inhibit the hypothalamus. And then the short loop is after the anterior pituitary, tropic hormones can go back to the hypothalamus in order to inhibit it. So, like you see here in negative feedback loops, any hormones released from either any glands or any organs in the future can go back to the origin organ, gland, etc., in order to inhibit it, to stop its stimulation, to stop more hormones from being produced. So here, negative feedback, A stimulates B, and then B can inhibit A. And then it could also be more complicated. A could stimulate B, stimulates C, stimulates D, and then D could inhibit A. But B could also inhibit A, and then C could also inhibit A. So it doesn't, so this would be in like D to A would be, or A to D, D to A would be a long negative feedback loop, while A to B, B to A, or even A to C, C to A would be a short negative feedback loop. So thyroid hormone and metabolic processes. So T3 and T4, which are over here, triiodothyronine and thyroxine. Definitely butchered that. But sorry about that. But T3 and T4, T3 is the hormone that's most produced by or T3 is hormone that's like it's only produced by 10%, but it's the most known in the body. So there's also a feedback loop. Um, and it's similar. So obviously here we're talking about the endocrine gland, but even in the thyroid gland, it's still the same process. A lot of these feedback loops have the same process. Hypothalamus releases TRH, or thyrotropin releasing hormone. This goes to your thyroid. This goes to your anterior pituitary, which releases TSH, your thyroid stimulating hormone. It goes to your thyroid gland, which then produces thyroxine. And then these could all go back to inhibit any of the original organs. So thyroid disorders. So hyperthyroidism and hypothyroidism. Hyperthyroidism is the overproduction of thyroxine. Hypothyroidism is the underproduction of thyroxine. And these two abnormalities do produce the same symptom, which are goiters, which, as you see in this lady, is like an abnormal mass that forms in the thyroid gland. So in both cases, the negative feedback loop becomes compromised, which causes an overstimulation of thyroxine or an understimulation of thyroxine. So in hyperthyroidism, which is an overstimulation of thyroxine, for some reason, your endocrine gland gets stimulated. As your endocrine gland gets stimulated, it produces so much thyroxine that it can't even inhibit any of the original organs. So the endocrine gland just keeps on getting stimulated, produces more and more thyroxine, and as a result, you have a huge mass in your thyroid gland. Hypothyroidism is there's an understimulation of thyroxine. When there's an understimulation of thyroxine, there's not enough thyroxine to help inhibit the anterior pituitary or help inhibit the hypothalamus at all. So if it can't inhibit the negative feedback loop, or if it can't inhibit any of the prior organs, the negative feedback loop does not work. And then obviously, once again, it produces the same effect where you have an increase in mass. Your thyroid gland has an increase in mass. So despite one being an overstimulation and one being an understimulation, they both cause the same effect, which is a goiter. So now we're going to talk about hormonal regulation of calcium. So when your calcium concentration is high, your thyroid secretes calcitonin. So calcitonin helps make sure that calcium from the blood could be brought in to the bone to help create new bones. And as a result, your blood calcium level lows. But if your calcium concentration level is low, then your parathyroid can secrete PTH, which activates osteoblasts and osteoclasts. So this helps shift calcium from your bone into the blood and also make sure your kidneys can help absorb more calcium and also your guts. It helps make sure that it could absorb more calcium. As a result, you have higher calcium levels in your blood. So, um, when your calcium concentration is high, the thyroid takes charge, but when your calcium concentration is low, the parathyroid takes charge. Now, glucose homeostasis, we're going to talk about insulin. I'm sure you guys know about diabetes, which obviously also plays a role in the insulin signaling pathway. So for glucose, so how insulin works is that you have a receptor in your cell. The receptor is part of your cell membrane. Insulin binds to the cell receptor, and then after it binds to the cell receptor, within the cell, vesicles release glucose transporters. So your vesicles release these purple glucose transporters, and then these go into the cell membrane. Now the glucose transporters can help take in glucose and then bring it inside the cell. So insulin is relatively like, or at least glucose homeostasis is relatively a simple-ish pathway. So once again, insulin binds to a receptor within in the cell membrane. This causes a vesicle to release glucose transporters. The transporters go within the cell membrane and it helps bring in glucose from the outside environment. Now we're going to talk about animal reproduction. So hormone independent versus hormone dependent characteristics. So hormone independent characteristics are your primary sexual characteristics, such as your X and Y chromosomes, your gonads, ovaries in females, testes in males, while your hormone dependent characteristics are known as your secondary sexual characteristics, and they include your genitalia, your brain, the hypothalamus, pituitary gland, other structures, and also different hormones as well. And the behavior you do, either just for humans or other animals, etc. Courting, mating, nest building, nursing, etc. So asexual versus sexual reproduction. So types of asexual reproduction, there's budding. So budding is you have meiosis. So there's growth through a meiotic cell division, and then it buds off the parent. So for example, like in hydras, the hydra has growth through meiotic cell division, and then there's like little buds that come off the parent. There's regeneration, such as in sea stars, for example. So damaged tissues could be replaced, even to the degree of growing a new limb. And there's nuclear fission, such as in sea anemones, and pathogenesis. So through sexual behavior, an organism could be produced from an unfertilized egg. An example is lizards or fish. So as you can see here, Hydra, these are budding. So through a meiotic cell division, these little buds eventually leave the original parent cell and then they grow into their own Hydra. While here, this is a sea star. These limbs are starting to regenerate. And then you have animals that have both sexual and asexual reproduction, and it depends on obviously the environment. So for aphids, In the summer is the perfect environment for them. So obviously, when it's the perfect environment, they want to produce as much of offspring as possible. So during the summer, they have asexual reproduction. So there, as you can see here, first instar, second, third, fourth, eventually live birth, and it continues on over the summer, several generations. But in the winter, conditions aren't necessarily ideal for asexual reproduction to occur. So during the winter, conditions aren't necessarily ideal. So during the winter, sexual reproduction occurs, where you have a female, sorry, you have a female, male, obviously, through sexual reproduction, they manage to produce an egg, which releases hatchlings. And by the time it's the summer, that hatchling could then go through my asexual reproduction, produce more and more aphids, and in time for the winter. So it's a cycle that goes on throughout the summer or winter. And then during reproduction, so for whiptail lizards, which we talked about was pathogenesis. So through sexual behavior, unfertilized eggs could still grow into different organisms. So here, uh, so in whiptail lizards, there are only females. So here you have a female lizard acting as a female. Here you have a female lizard acting as a male, and the the way a lizard acts depends on the hormone that's obvious the hormone level that obviously is most active during that time. So high estrogen leads to acting as a female, while high progesterone leads to acting as a male for these whiptail lizards. Now, sexual reproduction in animals, it consists of three main steps: gametogenesis, which is making your gametes, such as your sperm and eggs; spawning or mating with your partner, help bringing the gametes together; and then lastly, fertilization, which is when the zygote is formed. So, which statement about asexual reproduction is true? You guys could raise your hand or type in the chat, whichever one. All right, I'm seeing a lot of C, and yeah, the correct answer is C. Um, the main reason, I'm sure, like you guys know, single individuals is allowed to produce offspring because it's asexual. Like for example, for the aphids, you don't need another partner. You could create an identical copy of yourself. You can create an identical copy of yourself just by yourself. You don't need anybody else. However, at the same time, that's also one of the biggest quote unquote downfalls of um that's also one of the quote unquote downfalls of asexual reproduction, is that there's no genetic variety. So somebody asked, why is E not correct? Is because bacteria can also go through asexual reproduction, and there's some haploid bacteria, or there's some haploid organisms as well. Not all organisms are diploid, so you have to be careful about that. So if this was like, which statement about asexual reproduction in lizards, then obviously it could be E. But because there's bacteria or other organisms, it's not E. So now we're going to talk about gametogenesis. So spermatogenesis, which is helps produce sperm, and I don't know how Dr. Garofalo pronounces it, oogenesis, which produces eggs. So make sure you know the cycle and know what occurs at each step, because a question about what occurs during mitosis, what occurs during meiosis 1, meiosis 2 will definitely be on the exam. So definitely memorize like the terminology and what is at each step. So for example, you have a male germ cell, 2N, in mitosis. So obviously now we're at diploid. This is in the embryo. Then in an adult, the male germ cell becomes a spermatogonium. Then the spermatogonium becomes a primary spermatocyte. and then this is still in 2N. Then in the first meiotic division, it becomes a secondary spermatocyte, and now it's starting to become haploid. Then in the second meiotic division, the secondary spermatocyte becomes a spermatid, and then lastly, that spermatid eventually matures into sperm. So now it's officially a gamete. So once again, make sure you memorize what occurs in each division. So male germ cell, spermatogonium, is still diploid. Then the spermatogonium goes under mitosis again to become a primary spermatocyte. Once again, it's still diploid. Then in the first meiotic division, it becomes a secondary spermatocyte. Now it's haploid. Then it undergoes meiosis again, meiosis 2, becomes a spermatid. And then lastly, that spermatid matures into a sperm. What's the difference between diploid and haploid? So diploid and haploid, diploid is 2N, haploid is N, and that refers to the amount of genetic material within the actual cell. So diploid 2N, haploid N. So anything that undergoes, so in this case, because you have a 2N cell and it undergoes mitosis, it remains as 2N. But meiosis goes from diploid to haploid, goes from 2N to N. So during the first meiosis division, primary spermatocyte becomes a secondary spermatocyte. Now we're talking about oogenesis. So you have female germ cells. So female germ cells becomes a oogonium. Also, once again, mitosis 2N. Then it becomes a primary oocyte 2N. Then it becomes a secondary oocyte. So from primary to secondary, it goes from 2N to N. And then honestly, it's a similar process to sperm. So you could remember that for spermatogenesis as well. In spermatogenesis, primary spermatocyte to secondary spermatocyte, 2N to N. In this case as well, 2N to N. So at least the phrasing is like similar. Now, the secondary oocyte becomes a ootid after the second meiotic division. And then lastly, the ootid grows into an egg. And then the polar bodies degrade because the polar bodies form, but then eventually when the egg comes, the polar bodies degrade because they don't actually have enough genetic material to become an egg itself. So they just degrade off. So, knowledge check. Of the various stages of human life, the earliest time an arrested primary oocyte would resume meiosis would be. Um, in the meantime, I'm going to get water. So I'll give you guys like 10 seconds so you could answer in chat. Um, yeah. All right, I'm back. Sorry about that. I'll share my screen right now. 1 second. So when would meiosis occur during oogenesis? Is basically what the question's trying to ask. If you guys would like to respond in chat, or when do you think meiosis would occur? So I see some people replying C, some people saying A, one person said E. So we're kind of split. I'll give it like 10 more seconds. So I'll say the correct answer. The correct answer is actually Brandon with E. So the reason it's not C is because remember a primary oocyte, if we go back, oh, a primary oocyte is not an egg yet, so it can't get fertilized. Only an egg could get fertilized. The primary oocyte has to undergo meiosis in order to become an egg, and then that egg gets fertilized. So the primary oocyte cannot get fertilized. So um Brandon was correct that the correct answer was E at puberty, because at puberty, that's when females would start to uh female oocytes would start undergoing meiosis to help produce eggs. So Brandon was right that the correct answer is E. What are the first haploid germ cells produced in spermatogenesis? And this is where obviously oh sorry. This is where you have to remember the names and what occurs at each step. So what are the first haploid cells produced in spermatogenesis? What are they called? E. Yeah. All right. Good job. Yeah, the correct answer is E. That's once again a memorization based, and then it's also easy to know because even if they ask what are the first haploid germ cells produced in oogenesis, it would be um secondary oocytes because it's a similar naming cycle. When you go from primary to secondary, you go from 2N to N. And then lastly, which is released from the ovary at ovulation? So everyone's saying C, the ovum, which is the egg. All right, process of fertilization. So fertilization involves a complex series of events. So the sperm and the egg have to obviously recognize each other. So that's why they say you need for fertilization to occur, the two species have to be nearly chemically identical. We say nearly because, for example, that's why we have species such as like ligers, where even though it's not natural, theoretically it has occurred or can occur. So the sperm has to be activated, so it gains access to the cell membrane of the egg. Then the cell membrane of the egg fuses with the cell membrane of a single sperm. The egg blocks any entry of additional sperm. Only one sperm can fertilize one egg. That's it. And then the egg eventually becomes metabolically activated and stimulated to start development. And then lastly, the egg and sperm nuclei fuse, and then you create a diploid zygote cell, which then eventually becomes a living organism. So this is why I said species specificity. Sperms can only fertilize eggs of the same or really closely related species. Once again, we say really closely related because theoretically liars can occur, but even if it's not natural, but it can occur. There's cell specificity. So sperms fertilize eggs, not other cells they encounter on the way. So sperm can't fertilize. Oh, sorry. Uh, a sperm can't fertilize like the endometrium lining of the uterus. It can only fertilize an egg. And then number specificity, which is only one sperm can fertilize an egg. You can't have multiple sperm do it. So, where does production of sperm occur? So it obviously occurs in male reproductive organs. I would say there could be an image like this on the exam with like some of the words um like blanked out, or I wouldn't say yeah, blanked out essentially. So you'd have to know where each um thing would be located. So definitely try your best to memorize like where everything is located. So the oh sorry. So the seminiferous tubules which help produce sperm cells. So that is in the testes, and then this is just a cross section of it. So spermatogenesis takes place in the seminiferous tubules. So this is what we talked about, the actual process of spermatogenesis. So you have mitosis, for the germ cell, becomes a spermatogonium, then it undergoes mitosis again, becomes a primary spermatocyte, then it undergoes first meiosis, becomes a secondary spermatocyte, and it becomes haploid, undergoes meiosis 2, becomes a spermatid, and then lastly matures into sperm. And then Sertoli cells are important. Sertoli cells basically help cushion uh the sperm cells as spermatogenesis occurs. Also also provides nutrients and just helps make sure there's a proper environment for these sperm cells to eventually mature or spermatogenesis to occur. So Sertoli cells are really important as they basically once again help provide the right environment. So fertilization. So now we're going to talk about once again feedback loops again, and then we're going to talk about oh, sorry. So testosterone feedback inhibition of luteinizing hormone is stronger before puberty and after puberty. So after puberty, inhibition is weakened. So this stimulates Leydig cells. Leydig cells help also in the testes. Leydig cells help produce testosterone, and then testosterone as well as follicle stimulating hormone stimulates Sertoli cells, which we just discussed, to help produce the nutrients in order to sustain spermatogenesis. Then inhibin has a negative feedback loop to help inhibit FHA's release because obviously you don't want too much of something to occur. And then testosterone could inhibit GnRH, which stimulates the anterior pituitary, in which then stimulates obviously luteinizing hormone and FSH. So for oogenesis, like I mentioned before, what you'd have to do for the male reproductive tract, you also have to do for the female reproductive tract. It could be an image like this where you'd have to memorize each of the features and then obviously label it on an exam. And then this is where oogenesis occurs. So the ligament holds the ovary over here in place in the abdomen. So in the ligament, you have primary oocytes, and then primary oocytes eventually through meiosis 1 become secondary oocytes, and then the secondary oocytes eventually become ovum. And then the corpus luteum is important. The corpus luteum helps produce progesterone for females, and then progesterone is important because it helps progesterone helps with maturation of cells and also helps with fertilization of eggs during for sexual reproduction. And then when the corpus luteum disintegrates, that's when progesterone also levels go down. And when progesterone levels go down, that's when the uterine lining starts to shed. So that's after the 28-day cycle for oogenesis. So if you release one small egg, why do we have a few days period? So I'm not sure why it's a few days. I wouldn't know about that, but what I do know is that I know there's uh here. So after ovulation, the corpus luteum starts to disintegrate. So once the corpus luteum starts to disintegrate, that's around day 28. As you see here, look at progesterone. So progesterone increases, right? Progesterone is important because that's what helps with fertilization of the egg, and it also thickens the uterine lining for sexual reproduction. And that's why people say like around this time is technically like the right time for the chances of fertilization to occur at its strongest. However, at around here, day 28, that's when the corpus luteum disintegrates. When corpus luteum disintegrates, as you see here, progesterone takes like a nosedive. And when progesterone takes a nosedive, that's when your uterine lining starts to shed. So when your uterine lining starts to shed, that's what is a period. For females, I'm not sure why it's a few days. I'm assuming it has to wait until progesterone levels go eventually like go back up or reach a certain amount, reach like the threshold for the period to stop. So I'm not actually sure why it's a few days. Sorry about that. All right. So the follicle. So follicular cells proliferate in response to FSH stimulation, and they help provide nutritional support of the oocyte, and they secrete estrogen. So after ovulation, follicular cells become the corpus luteum, which secretes progesterone, which I was just talking about. And when there's not enough progesterone, a miscarriage happens because um, progesterone, obviously, like I mentioned, it helps provide support. And if you don't have enough progesterone, obviously you don't have the support for the fetus or etc. to occur. So miscarriage could happen. So negative feedback regulation occurs through most of the ovarian cycle, and then luteinizing hormones and FSH levels remain constant, like you see here. They remain relatively constant until like ovulation occurs. During that time, positive estrogen feedback enhances, so it starts to stimulate GnRH, luteinizing hormone, and FSH release in the middle of the ovarian cycle. So your luteinizing hormone surge occurs 2 to 3 days before ovulation, and then failure of positive feedback and luteinizing hormone surge prevents ovulation. So if you don't have that positive feedback loop, if so, for example, if estrogen doesn't stimulate luteinizing hormone and FSH and the positive feedback loop does not occur, then ovulation cannot occur. So ovulation over here, day 14, occurs because luteinizing hormone, like progesterone as well, but luteinizing hormone definitely, their hormone levels increase to such an extent where ovulation can occur. So here's another thing. Memorize these charts. I remember when I took the class, there was like a chart like this, and then the question was, what hormone is represented by this line, or what hormone is represented by this line? So definitely memorize the charts so that way you know, well, obviously what the hormones are and get the question right. And yeah, like I mentioned here, for this it would be easier because, like luteinizing hormone is the main hormone responsible for ovulation. And then if you don't have that positive feedback loop to help stimulate the pituitary gland to release luteinizing hormone and FSH, then ovulation cannot occur. So pregnancy. So over the course of pregnancy, human chorionic gonadotropin level are maintained while estrogen and progesterone levels increase steadily. And then the estrogen to progesterone ratio also increases slowly. So as you see here, months since start of last menstrual period, as a woman gets pregnant, estrogen and progesterone levels increase significantly. While at around like the third month, HCG also remains pretty constant. Like from the third month to the last month, it remains pretty constant. So estrogen is produced by your placenta, and then placenta is also what provides nutrients to the baby or to the fetus during pregnancy. Um, it also stimulates endometrial and mammary duct growth and suppresses prolactin and lactation, while progesterone is also produced by the placenta and the corpus luteum. It stimulates endometrial growth, suppresses luteinizing hormone and FSH release, and also suppresses myometrial contraction. So knowledge check. In women, a drug that blocks the luteinizing hormone surge would most immediately prevent What? B. Yeah, correct, B. Like that chart showed, day 14, that's when ovulation typically occurs, and that's due to the surge in luteinizing hormone. What two pituitary gonadotropins coordinate female reproductive events? Basically, what two are the main hormones responsible? We have B, someone said E, some people saying E. Oh, sorry. So the correct answer is E, um, luteinizing hormone and follicle stimulating hormone, because they're from the pituitary gland, pituitary gonadotropins. So while estrogen and progesterone, they're not necessarily produced from the pituitary gland. Um, they're if we go back, they're produced from the ovary. So the anterior pituitary gland produces luteinizing hormone and follicle stimulating hormone that stimulates the ovary, and then the ovary helps produce estrogen and progesterone. So estrogen and progesterone obviously does play a major role, like we discussed. It does play a major role in pregnancy, but it's not a pituitary gonadotropin. It's primarily E. Controlling fertility. Um, so this is just there's several methods of contraceptives in order to help control fertility. Um, Obviously, if you are unprotected, high rate of fertilization, high risk of pregnancy. Um, Also rhythm method, coitus interruptus. There's several methods. Um, Condoms, jellies, cervical caps. There's also hormone-based methods. So oral hormones. There are also non-oral hormones, which are like patches. Then there's also Plan B. So they all have varying success rates. And then typically like IUDs and there's also IUDs, a drug, mifepristone. Um, for these, you just have to know that there are lots of methods that help prevent or try its best to prevent pregnancy as much as possible. And then vasectomies and tubal ligations, vasectomies in males, tubal ligations in females. These are like what people say is like the bet, technically the best or highest success rate of preventing pregnancies. So now we're going to talk about animal development. Um, I believe after this, there's only one more chapter, so uh, stick through it. So, like I mentioned, so there are some concepts here. So here we're already starting at 42.2. That doesn't necessarily mean 42.1 is not important. It just means that 40 for this chapter, I wanted to start at 42.2 because that's where like the main ideas of the chapter is, but definitely still study what 42.1 is in the textbook or in the actual slides from Dr. Garofalo himself. So what is cleavage? So cleavage is after a zygote is formed. Cleavage is when there's early cell divisions. There's no cell growth happening, but the cell starts to divide and become a small ball of small cells. So eventually it cleaves into a blastula. The embryo becomes a blastula. And then the cells of a blastula are called blastomeres. And then a blastocele is a cavity within the actual blastula itself and is fluid filled. So the pattern of cleavage varies with the type of species. So for mammals and frogs, for humans, it's complete cleavage. It occurs in eggs that have little yolk. The blastomeres are similar in size for the most part, but in frogs, the vegetal pole has more yolk, so division is unequal, and daughter cells in the animal pole are smaller. While for humans, they're all relatively the same size. So, but even if it's not the same size, like in complete cleavage, what that means is you have a complete division of cells. Incomplete cleavage occurs in eggs where there's lots of yolk. So, for example, birds, fishes. So the cleavage furrows don't necessarily penetrate the egg completely. So you have, like you see here, an incomplete cleavage. It doesn't necessarily go through the entire cell. And lastly, there's discoidal cleavage, where the embryo forms as a blasted disc, like over here, and it sits on top of the yolk. Superficial cleavage is primarily found in insects, and it's a variation of incomplete cleavage. So the plasma membrane goes inwards around the nuclei. So mitosis occurs without cell division, and then a syncytium, a single cell, like here, these little white dots form, and then the nuclei migrate to the edge of the cell membrane, and then this creates a blastoderm surrounding the actual cell itself. So in mammals, fertilization occurs in the oviducts. So so here is where fertilization occurs. So here is obviously the ovary. It releases the egg. Sperm travels through here and then comes here. And that's where fertilization would occur. And then eventually the zygote would travel down the oviduct into the uterus. And that's when the zygote would start to mature. Oh, like it says here, site of fertilization, and then it slowly travels down into the uterus. So implantation occurs when the trophoblast adheres to the endometrium. So the endometrium is what we talked about is the uterine lining. So that's also what sheds during periods. So implantation that occurs oviduct is an ectotopic or tubal pregnancy, and then the zona pellucida, which it's not here, but the zona pellucida normally prevents implantation from occurring in the oviduct. And also, this is what we talked about where there's cell specificity. If we go back, the sperm travels through here, right? But the sperm does not fertilize any cells here, even though it's completely surrounded, because it is specified for an egg, which it'll meet in the oviduct. So the sperm will not fertilize the uterine lining cell. It'll only be here, even though it travels throughout everything. So gastrulation is you have movements of cells within the blastula. So you go from an embryo to a blastula. Oh, you go from a zygote to a blastula and now a gastrula. So gastrulation, massive movements of cells transform the blastula into an embryo that has multiple tissue layers and distinct axes. So in triploblastic cells, as you can see from the name triplo, tried 3, there are 3 germ layers, tissue layers that form ectoderm, endoderm, and then the mesoderm here. So the ectoderm is the outside layer, endoderm inside layer, and then mesoderm is the middle layer. Honestly, like you see ecto, endo, meso, it fits our human body. The endoderm becomes the lining of your digestive tract, your respiratory tract, thyroid, liver, etc. While your ectoderm is your outer layer, becomes your nervous system, your eyes, ears, and your skin as well. The mesoderm, which is the middle layer, it contributes tissues to many organs, which becomes your heart, blood vessels, and your muscles and bone. So here, for example, you have your blastophore, so your animal hemisphere at the top, vegetal hemisphere at the bottom. The vegetal hemisphere starts to flatten, and then as it flattens, it kind of pinches inward to form the archenteron. Other cells start to break free, becoming the primary mesenchyme, and then more cells break free, becoming the secondary mesenchyme, and then these attach to where the overlapping endoderm is here, and then it kind of forms like these little like you can kind of like it's not a filament, but you kind of think of it as a filament that attaches to it. Then the archenterum is elongated by the contraction of these filopodia, which is basically these long lines here. So they contract, so they bring the archenteron with it. Eventually, this causes the mouth to form at the very top. At the animal hemisphere, the mouth will form where the archenteron meets the ectoderm, while the blastopore will form the anus of the cell, which is at the vegetal hemisphere. So yeah, like it says here, archenteron cells extend, flatten, and migrate to form a long, thin tube. The mouth forms where the archenteron meets the ectoderm, so animal hemisphere, while the blastophore is the opening formed by the vegetal pole as the archenteron goes up, and that eventually becomes the anus. So gastrulation, once again, produces an embryo with three germ layers. Then there's organogenesis, which is your organs developing, and neurulation, which is initiation of the nervous system. So a group of cells, so here you have a specified ecto, meso, and endoderm. So a group of cells pass over the dorsal lip and then they become the chordam mesoderm. It's not necessarily that like this part, it's not necessarily as important to know, but all you have to know is that the notochord eventually forms. So the notochord forms and it provides support from the embryo and it causes the ectoderm to start forming the nervous system. So here, the ectoderm lying over the notochord, which is here, it thickens and starts forming the neural plate. Then the neural plate starts to fold, and then a deep groove forms. Then these folds start to fuse, forming the neural tube. And then the neural tube forms, and now you have a layer of ectoderm as well. So once again, your notochord, you have the neural tube at top. So neural crest cells, which are these here, they dissociate from the neural tube. So they're part of the they're part of the neural tube at first, but eventually they dissociate and they lead the development of connections between the brain and spinal cord and the rest of the body. So like we mentioned, the nervous system is throughout your entire body. It's throughout your hands, feet, etc., and also your brain and spinal cord. So that's what's caused by the neural crest. These help create those connections. And they also help create diverse structures such as your jaw, your skull, your face, smooth muscle, peripheral nerves, etc. Homeotic genes control body segmentation, all have a DNA sequence called the homeobox. And then invertebrates, Hox genes are homeotic genes that control differentiation along the anterior posterior body. And it goes in order. So, for example, here, your hindbrain is produced by HOXB1, while a HOXB9 over here, that helps form the spinal cord all the way down to the posterior side of the body. So like they said here, it's almost in order where at the 3' end, the genes closest to the 3' end are expressed in the anterior position, while those closest to the 5' end are in the posterior position. Yeah. So now we're going to talk about gestation. So pregnancy in humans is about 9 months and it's divided into trimesters. So in the first trimester, the embryo becomes a fetus. So by week 4, the heart starts to beat and limbs start to form by week 8. So this is what the embryo starts to look like within 4 weeks and the heart starts to beat. Then this is what it looks like after 8 weeks. So in 4 weeks, as you can see, it looks completely different. So, and like it says here, the first trimester is when rapid cell division and tissue differentiation occurs, but it's also the most scary time in the sense that the fetus is most susceptible to damage from radiation, drugs, chemicals, smoking, etc., that can cause birth defects. So that's why embryos could be damaged even before the mother realizes she's pregnant, because 8 weeks is only 2 months out of a whole 9 month span. So as a result, if the mother is not taking proper care of herself because she doesn't know that she's pregnant, the fetus could be susceptible to lots of pathogens, lots of disorders, etc. So in the second trimester, after 4 months, the limbs start to elongate, fingers, toes, and facial features form, and the nervous system starts to develop, which is what we were talking about with the notochord. And then in the third trimester, internal organs start to mature. And then lastly, birth occurs when the lungs are finally mature enough. And so the potential for serious effects from exposure to environmental factors exists throughout pregnancy. There are lots of factors such as protein malnutrition, alcohol, cigarette smoke that could also all result in low birth weight, mental disorders, other complications, etc. And that all could happen within the first 2 months without the mother even realizing she's pregnant. So now we're on the last chapter, which is the nervous system. So what are the major functions of the nervous system? So it helps cause interactions of the external environment and the internal environment. Everything that we touch, we feel, we think is caused by the nervous system and our reaction to our interactions with our external environment. So you have the sensory nervous system and your motor nervous system, which regulate the responses. We have autonomic control, which are reflexes. These are controls where you don't have to think about, they just happen. And then voluntary control, which is like me actually like moving my arm or my hand up and down right now. And it's all integrated with other organ systems and other organs that help regulate these systems and help maintain homeostasis. So organizations, so nervous systems are organized completely differently throughout lots of animals. So in here, in sea anemones, you have a nerve net. So that's only for simple behaviors to contract or relax. In earthworms, you have ganglia within each individual segment. So the ganglia in each segment actually help coordinate movement. And then the anterior ganglia here control more complex behavior. In a squid, you have more complex behaviors from neurons and specialized ganglia that help process and integrate information. And actually, the reason we know so much about action potentials is because of the squid's giant axon. So the axon of a squid is much larger than a human's, where you can actually experiment on it properly. So as a result, a lot of what we know about action potentials, the nervous system itself, comes from squids, thankfully. And lastly, you have humans. So the brain and the spinal cord make up the central nervous system, which makes sense. It's in the middle of your body, while the nerves that reach out to every other ligament or every other limb in your body is your peripheral nervous system. So like I said, central nervous system, brain, spinal cord. Peripheral nervous system consists of ganglia, nerves, and sensory receptors throughout your body. So sensory systems input. You have receptors. So for example, you have receptors on your fingers. Then you have afferent nerves. So you have afferent and efferent nerves, which we'll go over. Afferent you could think is approaching. So approaching the brain. Efferent nerves exiting the brain. So afferent nerves help send signals to the brain. It's approaching the brain. Motor systems are your output. You have higher and lower motor centers. You have motor nerves or efferent nerves that's exiting the brain and telling your limbs, etc., what to do. And then it's also your muscles and glands. Then you have your autonomic nervous system. Autonomic nervous system consists of your sympathetic nervous system, fight or flight. Once again, it's automatic. It's not something you necessarily think about. And then your parasympathetic nervous system, rest and digest. And then you also have higher association and integrative centers, which isn't as important. So the nervous system has two types of cells, neurons, which are your nerve cells. They help generate and conduct electrical signals or action potentials throughout the body. And then glia. So glia, there's macroglia, which help modulate neuronal activity. They help provide support for neurons. An example is oligodendrocytes, Schwann cells, which help produce myelin, and then astrocytes, which also kind of act as like the white blood cells of your brain. And then there's microglia as well. So there's small phagocytic cells derived from blood stem cells. And it's important to maintain your synapses. So obviously, the synapse is the area in between two neurons where the signal gets transmitted to. And so you need to maintain the synapse so that signals or the neurotransmitters can be properly transmitted from one neuron to another neuron.
So the structure of a neuron. I remember we looked at this briefly in the first chapter, but here we're looking at it more specifically. So here you have dendrites. So dendrites are the branches of a neuron which help receive information. Your soma, the cell body, which helps contain the nucleus. And then here is the axon. The axon is where the action potential is sent. So the action potential basically helps send a signal along. So the axon is where the action potential is sent throughout. Here you have the synapse. The synapse is the area in between the two neurons. So in the presynaptic cell, postsynaptic cell. And then that's where the neurotransmitters are released into, and they send a signal to the dendrites of the postsynaptic cell, and it continues on and on. So insulation. So Schwann cells help produce myelin. Myelin covers the axon. Myelin covers here. The long line you could see here. Myelin covers the axon, and that's important because you'll there's something known as saltatory conduction. So it helps the signal essentially jump from one node to another node to another node. You don't necessarily need lots of signals like going on throughout. It could jump from one node to another node, and it helps speed up the process. So as you can see here for neurons, there's lots of different types of there's lots of different what is it, morphology or forms of neurons that it could take. Bipolar cells, which are in your retina, they just help connect your photoreceptors to the ganglion cells. So all their only job is connection. So as a result, their function or their form is very simple. In the cerebral cortex, a pyramidal cell, it's much more complex. While in the cerebellum, a Purkinje cell, it's lots of dendrites. That one cell body has hundreds, if not thousands, of dendrites. So now we're going to talk about the action potential. So patch clamping is you have two pipettes. One pipette goes inside the neuron, one pipette goes outside the neuron, and you measure the electrochemical gradient both inside and outside the neuron. So you can measure the potential difference that occurs. So and then there's also patch clamping. Patch clamping, you have a pipette, you put it on a neuron, and you could suction off a part of the neuron. So as you suction off the part of the neuron, you could actually trace the ionic current that occurs within that suction, and that would be true for the entire neuron. So ionic gradients across the membrane. So you have a lot of potassium within the cell and then a lot of sodium outside the cell. That's pretty much the main thing you need to know. Like people say neurons are a salty banana. You have lots of salt, sodium outside the cell, and then bananas are known to have lots of potassium within the cell. So there's an electrical chemical gradient that occurs within the neuron. So opposite charges attract, like charges repel, and there's a net movement of particles from high to low. So like you see here, there's a concentration gradient. So a lot of people think, oh, why is there so much potassium within the cell and not outside the cell? Here's why. So like you see here, there's a lot of potassium within the cell, but not outside the cell. So what should happen is it evens out. However, you have to take into account the ionic concentration gradient or the voltage gradient. Because there's so many, because there's so many negatively charged macromolecules within the cell, you need a high concentration of potassium on the inside. So the electrical chemical gradient takes precedence over the concentration gradient. So once again, there's a lot of potassium in the cell, so you'd expect it to even out. But because you have so many negatively charged macromolecules within the cell, that is more important because you need to even out all the negatively charged molecules within the cell. So the electrical chemical gradient takes precedence, so you bring in a lot more potassium within the cell. So this is pretty easy. What's the chemical environment of the cell during resting membrane potential? What's the intracellular and extracellular environment like? Someone said D. D. Someone said A, D. So the correct answer is D. So A is partially true. You do have high potassium within the cell, but you don't have low sodium outside the cell. If we go back, see here, you have a lot of sodium outside the cell. So A is partially true, but D is the correct answer. High potassium within the cell, high sodium outside the cell. So now we're going to talk about the distribution of ions across the cell membrane. We're almost done, guys. So ion pumps use ATP or energy to help transport ions across the concentration gradient. So you have the sodium potassium pump, 3 sodium out, 2 potassium in. So the main function is to keep intracellular sodium low. You don't want sodium within the cell, you want sodium outside the cell at resting membrane potential, obviously. So that's the main point of the sodium potassium pump. Takes 3 sodium out and brings in 2 potassium. And then you have 3 types of gated channels: ligand gated channels, voltage gated channels, and mechanically gated channels. So here we're going to talk about the actual action potential itself. At resting membrane potential, you have potassium channels open where potassium could freely go through. Then only leak potassium channels are open. Voltage gated channels are closed because you haven't reached the correct voltage yet, and chemically gated channels are also closed. Then depolarization occurs. Once the membrane reaches threshold, so I'm sure you guys have heard that action potentials are all or nothing, and that's true. You have to reach threshold. Once it reaches threshold, an action potential will definitely occur. So once threshold is reached, an action potential occurs where voltage gated sodium channels open. When voltage gated sodium channels open, sodium goes through the channel. And I believe someone mentioned that in chat. High sodium outside until the channel's open. Yep. Um, once the threshold is released, voltage gated sodium channels open. Now all the sodium is rushing into the cell. And as you imagine, you have a lot of positive ions rushing into the cell. What happens? Depolarization, which is your membrane becomes extremely positive. Now, eventually, as your membrane becomes extremely positive, now hyperpolarization needs to occur. Hyperpolarization is when it eventually goes back down to your resting membrane potential. So your chemically gated potassium channels open, potassium starts to flow in and out. It starts to flow in and out, and eventually the sodium potassium pump also takes out 3 sodium, brings in 2 potassium, and you reach resting membrane potential. So, as we described here, resting potential, only leak channels are open. Potassium goes in and out, everything's fine. 2 here, you reach threshold. Here, -50 is threshold. And then around people believe around -70 is typically where resting is. So in this case, -50 is threshold. Once threshold is released, voltage gated sodium channels start to open. As they start to open, sodium rushes in. As more sodium rushes in, more voltage gated sodium channels open, more sodium rushes in. Eventually, the chemically gated potassium channels open and the voltage gated sodium channels close. So the voltage gated sodium channels close, more sodium cannot go in. But the chemically gated potassium channels open where potassium goes out. So now as potassium ions go out, the cell goes negative. And undershoot is basically when the cell goes more negative than normal, but that's because you don't want another action potential. It's like a preventative measure to make sure another action potential does not occur. That's not necessary. So eventually then the sodium potassium pump comes into play again to help bring the cell back to resting membrane potential. So propagation of the action potential. So this is what we're talking about with the voltage clamp. You have a pipette inside the axon, outside the axon. It just measures it. And as you see here, in point A, you have an action potential. and then point B, it goes through. It propagates throughout the axon. So here, sodium ions enter at point A over here at time 1. So this yellow means that an action potential is occurring. And then they flow down the axon toward point B. And eventually, over here, the voltage-gated sodium channels close. But here, the voltage-gated sodium channels start to open. And now you have an action potential at point B. So all this is saying, it propagates throughout the axon. You'll go from point A to point B. And saltatory conduction in myelinated cells, this is what we were talking about. Let's say this is point A, sodium channels open, you have an action potential here. This the myelinated Schwann cells help spread the current so that this node already starts to reach threshold. Now that the second node already starts to reach threshold, it's much easier for it to reach an action potential. So it reaches the action potential and the process continues on and on. That's why we say it jumps from node to node. This node already has the action potential. The myelinated cells spread the current so that the other node is already essentially prepared. So it reaches threshold, it's prepared for the action potential to occur once the sodium ions travel down. Synaptic transmission. So this is what occurs from the presynaptic neuron to the postsynaptic neuron. Calcium goes into the presynaptic cell. When calcium goes into the axon terminal of the presynaptic cell, this causes neurotransmitter, in this case acetylcholine, this causes neurotransmitters to bind to the membrane and then release. This causes neurotransmitters in vesicles to bind to the membrane and release the neurotransmitter into the synapse. Once it releases the neurotransmitter into the synapse, it binds to receptors in the postsynaptic cell. Once it binds to receptors in the postsynaptic cell, as you can see here, sodium starts rushing in and another action potential is caused in the next cell because obviously you want the process to continue. You want the chemical signal to continue until it reaches the brain or until it reaches the muscle, etc. So spatial summation versus temporal summation. All you need to know about this is that spatial summation, several potentials arrive at the axon at the same exact time. So several different potentials, they arrive at the axon hillock at the same exact time, while temporal summation is the same synapse. There's several postsynaptic potentials at the same synapse so that they could be summed into one big potential. So spatial summation, different potentials, but they arrive at the same time at the axon hillock, while temporal summation, same synapse, but you have a lot of potentials, so much so that they could just be summed up into one big potential. And then this is the last slide. This is just an overview of the entire nervous system of what occurs, for example, during the reflex. A hammer taps the tendon in your knee. Now these receptors fire action potentials, afferent neurons approaching the brain. So the action potentials go through and then they reach the brain and they reach the ventral horn of the spinal cord. Then it sends a signal to a motor neuron here. Then the motor neuron sends a signal to the muscle through efferent nerves exiting the brain. It conducts action potentials. It continues conducting action potentials, but now towards the muscle. It conducts action potentials towards the muscle, causing a contraction, and it inhibits the antagonistic muscle. So an antagonistic muscle is basically the muscle that causes some relaxation. So it inhibits firing an antagonistic muscle, but conducts action potential in the extensor muscle, causing contraction, and now the leg extends, and that's the entire action potential pathway. So that's the end of the exam 3 review. If you guys haven't, please fill out these QR codes of the attendance feedback. The tutors, me, we'd really appreciate, what we could do better, how we've done, etc. If you guys have any questions, I'm here until 5. Please ask any questions, general, how to do well in class, study, etc. You're welcome. Yeah, ask any questions. I'm open to everything. I took this class with Dr. Fat, Dr. Garofalo. I know them very well. So, or if you don't have any questions and feel confident, I guess that's a good thing. Did my job somewhat well. Let me put on the light actually. Give me 1 second. The pinching thing I mentioned about the neuron. If it's not on the actual slides, oh yeah, here. Uh, oh, give me 1 second.
I'm sorry to interrupt, but yeah, as Sharnom mentioned, please make sure you fill out the feedback form. It's a great help to us to improving the review sessions and helping support students' needs. So thank you so much.
So with this pinching thing, if it's not on the actual slides itself, then it's not necessarily important. This is just a way they measured ionic currents because um it just helps to obviously measure. There's multiple ways to measure ionic currents. This is just one way. So if it's not necessarily on the slides itself, it's fine. Um, how should you study for exam? What helped me the most? So what I'd say what needs to be applied when these is there going to be a curve for biology 105? There is not a curve at the end of the semester. At best, maybe like a singular point, maybe two, but do not rely on it. It changes depending on the semester. At most, like I said, at most it'll be 2 points. It's not yeah, don't rely on it, basically. It won't save your grade. Um No, Bio 105 does not have a curve. Sorry. In terms of best methods to study for the exam, I definitely say study from the slides for sure. Achieve helps, but the slides are the best because I think Dr. Fath or Dr. Garofalo themselves have mentioned so many times that like whatever is on the test is on the exams. The problem is, like I'm sure you guys know, I condensed a lot of material and there's a lot of material I didn't necessarily like cover, but there's still a lot more material than this. Like each of his chapters are around 50 to 60 slides. So the problem is that obviously there's a lot of information to memorize. The best way to study is, like I said, study from the slides as best as you can. What I do is that in class, there's times where Dr. Garofalo or Dr. Fath, they emphasize certain slides. Or let's say there's a slide where they go over it for several minutes, while there's another slide that may have like lots of information, but they only go over it for like 30 seconds and then skip past it. In those cases, like it's easy to tell from Dr. Garofalo or Dr. Fath that they're not going to focus on that slide with a lot of information as much. That one slide that they spent several minutes talking about, like those are the main ones. But everything they talk about is from the slides. As specific as it is or as general as it is, it's from the slides itself. Now, studying from the textbook, um, I wouldn't recommend studying from the textbook as much due to the fact that the textbook has so much information. So as students, obviously, there's so much that you already need to memorize from the slides itself, and the textbook has even more information that you'd need to memorize. So in those cases, the textbook could actually hinder your score or what you would achieve in the class because there's just so much more info you need to memorize that'll just like cover up what you're thinking about. So studying from the slides is the big thing. If anyone records lecture, I know Dr. Garofalo or Dr. Fath are fine with audio recordings. If anyone records lecture, definitely try your best to like re-listen to it at like 1.5 times speed or something, because that way you could also hear like when they talk about something for a long time. Recording lectures is definitely something that helped me a lot. And another thing I'd like to recommend quickly, sorry, Drew. Another thing I'd like to quickly recommend is that focus on your lab as well, because no other class, at least within the major science fields, other than bio, has lab be such a major portion of your actual grade. Lab is 40% of your grade. And so if you do really well in lab, and let's say you're not doing as well in lecture, your lab can help boost your average significantly. And so focus on lab as best as you can. Try to submit your lab reports, do well on quizzes, because there's lots of people where, like I mentioned, they may not do as well in lecture, but thanks to lab, they help boost their average as much as possible. So 40% is a significant portion of your grade. Furthermore, do your homework on time. I believe homework is the same as like one test grade. Um, so if you think about homework as like all of your homework as one test grade, you could consider like, oh, I got 100 on one test. So definitely do your homework on time. Make sure you do that perfectly. Yeah, study from slides. focus on lab. If you have recordings, listen to recordings. I wouldn't recommend studying from the textbook or Achieve. And maybe I wouldn't recommend solely studying from practice exams as well. I know a lot of students have practice exams, but that's their only method of studying. I wouldn't recommend doing that. I'd recommend going over the slides first and maybe like a day or two before the exam, then you only focus on practice exams because that could also help test how much you really know. And also, like after you get like, let's say you get a certain question wrong, you can know, okay, maybe I need to focus on this chapter more, or I need to focus on that chapter more, or oh, I did really well in this chapter, I don't need to focus on it as much. So don't just solely study from practice exams. Leave your practice exams to like your last day or your last 2 days. Now, zygote to blastula. Give me 1 second. Sorry, they're all in one major. Oh wait, is it chapter 42? Yeah, so zygote to blastula, it's not as like there's nothing super complicated that you need to memorize. The main thing is that the zygote undergoes cleavage, so no cell growth occurs during this time, but the zygote itself becomes a solid ball of small cells. It just divides. And then the type of cleavage, obviously, it depends on the type of animal, but the zygote starts to cleave, and eventually, through more and more cleavage, it becomes a blastula. So the embryo becomes a blastula. Each individual cell is called a blastomere, and then within the blastula, there's a fluid-filled cavity that's called a blastocele. That's pretty much the main thing you need to know, at least for this process. And then there's different types of cleavage that occur. Humans, frogs, and I'm assuming humans and frogs, complete cleavage, where there's actual clear divisions throughout the actual blastula. And it may not be so complete cleavage, it may not be equal. So that's the thing. There could be unequal or they could be unequal, like in frogs, like you may have like bigger cells at the vegetal pole, smaller cells at the animal pole. However, it's still complete cleavage because cleavage occurs throughout the entire blastula. Then there's incomplete cleavage, such as in birds or fishes, where cleavage does not go throughout the entire mass. And then discoidal cleavage, where the embryo forms a blasted disc that sits upon the yolk. So that's pretty much the majority of what you need to know for incomplete versus complete cleavage. You're welcome. 5 minutes left. If anyone has any more questions or anything, maybe how to do well in lab, etc., let me know. Otherwise, also, I know a lot of people left, but thank you all for being here. Please fill out the form if you can. It would be really helpful as I speed through it now. Yeah, thank you all for being here. Will the recording be posted? Arena, I'm not. Do you think you can answer that? I'm not actually sure.
Yeah, so it should be we could post the link, yeah.
Okay. And then what are some key points of animal reproduction? So the key points of animal reproduction, in my opinion, definitely memorize what spermatogenesis is, oogenesis is. Those are like the fundamentals of how fertilization occurs. And then definitely memorize the different types of reproduction that can occur: asexual reproduction, sexual reproduction, the advantages and disadvantages to each. Asexual advantage is that it's super quick, and that you don't need another partner, but there's no genetic diversity. Sexual takes time and you need another partner, but you have genetic diversity, etc. Um, you're welcome. Um, are there any memorization tactics for all the hormones to memorize? Unfortunately, I don't necessarily have any tactics, but the good thing is I know I had that big graph image of like all the hormones. The thing about that is a lot of those hormones that are talked about, you don't necessarily need to memorize it for that one chapter because a lot of those hormones that I talked about are used in other chapters. For example, we talked about FSH, luteinizing hormone, estrogen, progesterone. Like these hormones are all throughout. Go over ACTH. By ACTH, do you mean acetylcholine or which hormone are you talking about, Drew? The cortisol one for stress. Do you remember what chapter? Did I discuss it? The endocrine system. Give me 1 second. Let me try to find it. Oh, let's see if it's in my slides. You're welcome. Um, chapter 39. I'm sorry, I'm not sure if I have that in my slides. Is there anything specific that you'd want to know about it? Like, do you remember? Like what specifically, if you remember, like I'll try my best. Pituitary releases. Pituitary controls cortisol release by acetylcholine, but then he said acetylcholine is controlled by CRH. Uh, is by CRH, do you mean is that a hormone or? I mean, that could also be related to, okay, that could also be related to feedback loop. So the pituitary, which is the gland, obviously releases a lot of hormones. So I'm assuming that in this case, it release what he's talking about at first is that oh adrenocorticotropic hormone. Sorry, not acetylcholine. Oh yeah, here. Adenocorticotropic hormone. So yeah, the pituitary gland releases ACTH. So this stimulates the adrenal cortex, and then obviously that also could like you see here, give me 1 second. Like you see here, pituitary releases ACTH, which then instead of endocrinal gland, let's say this is the adrenal gland, and then here would probably be cortisol. And so there could also be negative feedback loops where the cortisol could also cause an inhibitory response to the anterior pituitary so that ACTH isn't released later on. I'm not sure if that's specifically what helps or um it doesn't necessarily release it twice. It's the fact that it's a feedback loop, remember? So let's say the anterior pituitary. So ACTH is only released from the anterior pituitary. So ACTH is released, yeah, it's specific to feedback. So ACTH releases the anterior pituitary releases ACPH, which then goes to the adrenal gland, which then releases further hormones. But these further hormones can cause a negative feedback loop to the anterior pituitary so that other hormones are not released. So like here, instead of the endocrine gland, if it was adrenal cortex and this is cortisol, it could cause a negative feedback loop back to the anterior pituitary. Yeah, no problem. No problem. And if I didn't say it already, good luck everyone on your exam next week.